Hello students, today we are going to start nucleic acids that composed of different types of subunits, that is nucleotides and nucleosides, and individual components which are responsible for the constructions of nucleic acid. Myself Dr. Suresh Kumbar, and today's class we are begin with the nucleic acids. First we will see the definition of nucleic acids. Nucleic acids are the polymers of nucleotides. Right, these are the polymers of nucleotides composed of nucleotides, means nucleic acids composed of nucleotides. First we will understand the difference between nucleic acids and nucleotides, because nucleic acids are formed by the repeating units of the nucleotides. So we are going to study each and the individual component of the nucleotides, and we are going to study nucleoside and nucleotide. So what are the nucleosides are it is a basic unit which is composed of sugar and base. Okay, that the structures of sugars and base we are going to see in this video only, but understand the difference between nucleosides and nucleotide. Sugar and base, that is nucleoside. Nucleotide, that is sugar base is common between nucleoside and nucleotide, and extra phosphate group, right? Phosphate group. The overall combination sugar base and phosphate group makes nucleotides, and nucleic acids are the repeating units where nucleotides forms the nucleic acid. 1, 2, 3, likewise so many nucleotide forms the nucleic acid. It is a polymeric chain, and there is a phosphodiester linkage between, means two, suppose this is one nucleotide and this is second nucleotide, which are linked by phosphodiester linkage. The linkage between two nucleotides, it is of phosphodiester linkage. Right, what are nucleotides and nucleosides? So nucleotide, which is composed of three basic structures, that is nitrogenous base, pentose sugar, and phosphate group. And as of we know, nucleoside, which is composed of nitrogenous base and pentose sugar. So these two common things are same in both, means nitrogenous base and pentose sugar both are common in nucleoside and nucleotide. And tide means 3. How to remember this nucleotide composed of 3 units? Tide 3. The third one is phosphate group. Phosphate group is the third component of the nucleotide. Then now we are going to study individual components of the nucleotide. Nucleotide, which is composed of nitrogenous base. So nitrogenous base is of two types. One is purine and second one is pyrimidine. Both are heterocyclic compounds which composed of nitrogens. So based on the number of nitrogens, we are classified two nitrogenous bases. One is purine and second one is pyrimidine. So what is purine then? So purine, it is a 4 nitrogen containing heterocyclic compound, is also having fused ring structure. Means fused means two ring structures are fused each other to form a purine, whereas in overall structure it contains 4 nitrogen, whereas pyrimidine, which is having two nitrogens present in the 6-membered aromatic ring, means here only one ring is present, whereas in the case of purines there are two rings, means fused ring structure, one ring, two ring, and 4 nitrogen, whereas 2 nitrogen in pyrimidine. Now we are going to draw the basic structures of purine. Right, purine, which is composed of 4 nitrogen and fused ring. So fused ring, which is having 6-membered followed by 5-membered. Two rings fused each other to form a purines. Okay, here we can see 4 nitrogen, 1, 2, 3, and 4 nitrogen, and one is 6-membered ring, this is 6-membered ring, and this is 5-membered ring, fused each other to form a purine. Whereas in the case of purine, which is composed of 1 and 2 nitrogen and simple aromatic ring, whereas 2 carbons replaced by 2 nitrogens. Now we are going to see the details of purine structures, which is composed of 2 types of purine. One is adenine, also called as A, and second one is guanine, also called as G. In short form, we call it as A and G. Adenine, which is having basic structure of purine, but there is an extra amine group, right? And what are the difference between adenine and guanine? In place of amine group, there is a replacement with carbonyl group, and at other alternative place we have amine group. Means in adenine, with respect to purine, if you want to make adenine, purine plus amine group, that is adenine, and if you want to make guanine, purine plus C double bond O plus NH2, these two extra components present in the purine, that forms the guanine, right? Adenine and guanine. Whereas in the case of pyrimidine, we have three different structures, namely thymine, cytosine and uracil. Thymine, which is composed of, means in all the three cases, what are the common things? These are the common things. C double bond O, carbonyl groups are the common things, right, with respect to pyrimidine. And second thing, there are, what are the differences? Amine ring present in the cytosine, whereas C double bond O present in uracil and thymine. Okay, to make cytosine, what are the components? We need to add NH2 plus C double bond O. Here 2 C double bond O, whereas in the case of thymine, 2 C double bond O plus 1 CH3 group. What are the differences between these three? All these three structures having C double bond O common at this place, followed by amine in cytosine, C double bond O in uracil and thymine, and extra methyl group in thymine as you compare to uracil. This is the biggest difference between cytosine and uracil. As we know, thymine present only in DNA and uracil present only in RNA, right? Now we are going to second part, that is pentose sugar. Pentose sugar is of two types, one is ribose and second one is deoxyribose. We know that ribose sugar present only in RNA, whereas deoxyribose sugar present in DNA, right? We will study its structures. Ribose sugar means pentose sugar, which is composed of 5-membered ring. It is pent means 5-membered ring structure, out of which one of the atom is oxygen. 5-membered ring with oxygen, and all the basic structures of the basic components of the sugar in it. And deoxyribose sugar, whereas it is also composed of 5-membered ring with oxygen, but one change, that is at second place there is no oxygen. Deoxy means, D means removal of oxygen, that is a oxygen, removal of oxygen from the ribose sugar. Ribose sugar contains oxygen at second place, but in deoxyribose sugar there is a no oxygen at second place. And third component of the nucleotide, that is a phosphate group, which is responsible for the phosphodiester linkage, right? Nucleotide, which composed of nitrogenous base pentose sugar phosphate group, whereas nucleoside composed of nitrogenous base and pentose sugar, right? This is a basic example for the nucleotide, which is composed of, this is nitrogenous base, pentose sugar, and this is phosphate group, right? Likewise, we have nucleoside, only 2 units, nitrogenous base and pentose sugar. Okay, now I am going to show you how to draw the individual components of the purines and pyrimidine. First I will start with purine. Okay, as we know, purine, which is having 4 nitrogen and fused ring structure. First we will draw the basic structure of the purine. First 6-membered ring, with alternative nitrogen. Don't draw double bonds, just draw single bonds, and we will make its valency later, right? Double bond, alternative double bond, here double bond, and there is no double bond here, so it is H, 4 nitrogen, right? Now we will draw true structure for these purine nitrogenous bases. Okay, first one is adenine, and we know adenine and guanine are the derivatives of the purine, so it is very easy for us to draw the structures of purine and adenine and guanine. First we will draw the structures of adenine, nitrogen, skeleton structure first we will draw, then followed by addition of double bond, means placement of double bonds. We have drawn the basic structure of purine, whereas alternative double bonds present, and there is an additionally you can see single bond amine group, NH2 group. This group is present extra in adenine, also called as A, whereas in the case of guanine we will draw again basic structure of pyrimidine. First we will draw structure, then we will place the double bonds, right? So there is a double bond here, double bond and double bond here, and one NH2, this is NH. What are the difference between these two? One extra double bond O, and this is single bond NH2. These are the two extra components present in guanine, so we call it as a guanine, also called as G, guanine G, right? Likewise you have to practice the structures. Now we are going to draw the structures of pyrimidine. Basic structure contains 2 nitrogens and 6-membered aromatic ring, right? That we are going to draw here. 6-membered ring first as like benzene skeleton, followed by 2 nitrogen at alternative positions, and double bond, right? Double bond at alternative position. This is the basic structure of pyrimidine. Now we are going to draw the structures, 3 different structures of pyrimidine. One is cytosine, also called as C. Second one is uracil. Third one is thymine, right? Uracil is also called as U, whereas thymine is also called as a T, right? First we will draw the structures. Basic structure, that is nitrogen, 6-membered ring. Then what are the functional groups present? There are common, that is C double bond O, and one amine group at this position, and we will place the double bonds here. It becomes NH, right? What are the variations with respect to pyrimidine? That is amine group and C double bond O, right? We will draw the structure of uracil. Again basic structure, nitrogen, 6-membered ring, pyrimidine, and C double bond O is present, another C double bond O, and we will place the double bonds where only one double bond, and this becomes NH and this becomes NH. And this is uracil present only in RNA. What are the difference between pyrimidine? pyrimidine, and uracil, that is extra this C double bond O present, means as in the case of cytosine, C double bond O is at that place and there is a one more C double bond O. Amine is replaced by C double bond O. And in the thymine structure we will draw basic structure, that is, and C double bond O, C double bond O, and one more methyl group at this place and double bond and NH. The difference is C double bond O as it is in the, as we compare uracil and thymine, and extra methyl group. So this present in DNA. This is present only in DNA. This thymine is also called as a 5-methyl uracil because basic structure is that of uracil, only methyl group present in 5th position, extra methyl group in 5th position. Now we are going to draw the structures of pentose sugar. Pentose sugar, which is composed of 5-membered ring. Pent means 5-membered ring and with oxygen as an heteroatom. Okay, we will draw the structure. First we will draw pentagon. 1, 2, 3, 4 and connection. 5-membered ring and oxygen as one of the member in the 5-membered ring system. Okay, pentose sugar, and we will draw 4 vertical lines at junctions. 1, 2, 3, 4 vertical lines, and we will place the functional groups. OH at above at first carbon, at second carbon OH at below, at third carbon OH at below, at fourth carbon above CH2OH, and remaining places we will write hydrogen, and hydrogen. This is a structure of pentose sugar. We call it as a pentose sugar. We need to draw carefully pentose sugar, and this is also called as a ribose sugar. This is a structure of ribose sugar, right? In other side we are going to draw the deoxyribose sugar. We know the difference between ribose sugar and deoxyribose sugar, that is removal of oxygen on second carbon. Okay, first we will draw basic structure. Oxygen, 5-membered ring, right? 4 vertical lines, 1, 2, 3, 4, numbering 1, 2, 3, 4. Oxygen is 5, no problem. And what are the functional groups we are going to draw? Oxygen hydroxy group, only hydrogen here. Hydroxy group CH2OH, right? And in the remaining places what we need to add? Hydrogen, right? So what is the major difference? That is, here we can see in the ribose sugar hydroxy group is present, whereas in the case of deoxyribose sugar only hydrogen is present. That's why it is called as a deoxyribose sugar, right? Thus we can draw the structures of any given nitrogenous base and pentose sugar. Don't worry about the question. First we will understand and we will practice to draw the structures, so we can be able to draw any given structures or any given component of the nucleic acid. So hit the like button.